Hi, good afternoon. So today I'm going to present to you a part of my PhD work that's focusing on using different phage proteins for detection and therapy of Pseudomonas aeruginosa. So as you know, antibiotic resistance is a major public health problem worldwide, and one of the concerning pathogens is Pseudomonas aeruginosa. This gram-negative bacteria has become resistant and is classified as a high-priority pathogen for the development of new antimicrobials. Bacteriophages and their proteins have shown incredible results, and my focus in this work is on endolysins and receptor binding proteins. As you have heard today in a previous talk, endolysins are enzymes that degrade the peptidoglycan of the bacterial cell wall. And they have been used successfully against gram-positive bacteria. However, their use against gram-negative bacteria is more challenging due to the presence of an outer membrane that blocks the access of this endolysin to the peptidoglycan. On the other hand, we have the receptor binding proteins, or RBPs for short, which are known for their high specificity in binding and interacting with the host. Therefore, in this work, we characterized two newly isolated phages for Pseudomonas aeruginosa, along with their endolysins and RBPs. Our goal was to assess their potential to be used as either therapeutic or diagnostic tools for Pseudomonas aeruginosa in an attempt to fight the antibiotic resistance crisis. So we started with phage characterization. In the efficiency of plating assays, our phages were tested against 42 clinical isolates of Pseudomonas aeruginosa plus 6 reference strains of the same pathogen. And our phages detected more than 50% of these strains with high yields. On the stability assays, both phages were stable at a wide range of temperatures and pH, and more importantly, they were stable at physiological conditions. Therefore, they should be applicable in therapeutical settings. Transmission electron microscopy revealed that both phages have a myovirus morphotype with long and contractile tails. Genome annotation was performed using both sequence and structure homology-based tools, and it was possible to identify clusters coding for structural proteins and for lytic proteins. We will start with the lytic proteins of these phages, and it was possible to identify one endolysin for each of the phages. These two endolysins belong to the lysozyme-like superfamily, and they were cloned and expressed in E. coli. Their functional assay was performed against outer membrane permeabilized PA1 cells, which is a reference strain for Pseudomonas, and also other clinical strains. After incubation of these spot assays for 2 hours, we observed clear halos that indicated cell lysis. Therefore, we could conclude that we had successfully isolated and produced functional endolysins targeting Pseudomonas aeruginosa. Focusing now on the structural proteins, despite our best efforts, it was possible to identify only one RBP for one of the phages. This RBP is located at the end of the structural cluster and has homology to a domain that is present in other RBPs from other species. This RBP was cloned fused with the green fluorescent protein and then expressed in E. coli. The functional assays were performed using this purified fusion protein, and it was incubated with PA1 cells for 30 minutes. The cells were washed and were visualized under fluorescence microscopy. The results that we saw are presented in figure 8, and what we can see is that all PA1 cells are outlined with a green color. This is happening because the RBP is binding to the receptors in the bacterial cell wall, and the fusion tag GFP is producing fluorescence. Therefore, we could conclude that we have successfully identified and isolated a functional RBP with a high specificity for Pseudomonas aeruginosa. Now, I talked to you about two kinds of phage proteins. They have good applications, but they are different. Vandalizins, because they lyse the host cell, and in here we are talking about Pseudomonas aeruginosa, gram-negative bacteria. If they are combined with outer membrane permeabilizers, they could be developed into tools for the control of this pathogen and the control of bacterial infections. The RBPs, due to their high specificity in binding to this pathogen, can be developed into fast, reliable, and very specific detection tools that could be used for the diagnosis of the bacterial infections. Therefore, we can say that with the identification and of two functional analyzins and one functional RBP for Pseudomonas aeruginosa, we have expanded the arsenal of phage-based therapeutics and detection tools to control this pathogen. Thank you very much for your attention. I would also just like to thank my supervisors, Dr. Silvio Santos, Professor Joana Azeredo, Professor Yves Briers, and my colleagues at the Azeredo Lab and LAB. Thank you.